Okay. So today I shall talk on this nanotechnology-based drug delivery systems. At least the last few decades, at least 2-3 decades, this age is going on this nanotechnology, nanoparticles, and the products from this the nanotechnology. This already this matter is introduced by your chief guest, Dr. AICTE, very reputed persons. He's our senior person also, and this Madam Vice Chancellor, she also talked something about this the nanotechnology. Then the drug delivery systems. This is another recent area, and this drug delivery systems gets a lot of importance today, particularly when our basic theme is the safety and efficacy of the drug. When you consider the maximum efficacy of the drug and the maximum safety, that means the hazard should be less, side effects should be less, then this the drug delivery system has to be improved. If I give a little background that this how this nanoparticle or nanotechnology has come recently, before that, how the drug delivery systems have developed. I think in this beginning of this this ATAL program, your FDP programs, the participants will be aware, and so that they speak up gradually. This the at least last 70 years from 1947 or 1950 onwards, this how people think about the drugs, the products, the delivery systems, and its effectiveness. I will give you initially a little about this the delivery systems, and then we will come to this the nanoparticles and its advantages, disadvantages, and the different type of these nanoparticles. Then we'll describe also a research paper on this one. So, dear participants, as you know that the particularly the 20th century is a very revolutionary in the drugs and pharmaceuticals, particularly its delivery systems. As you know that this the first degree education started in 1932, Drugs and Cosmetic Act 1940, Pharmacy Act 1948, and then Pharmacy Council of India, and this education regulations, which is revised every 10 years, and then the AICTE come into the pictures. Then there's a lot of change in educations, and as well as in the pharmacy profession also. If you look that this before 1940, we said that this it was the earlier age, 1940 to 70, it was industrial age, and 1970 to 1990, it is a patient care age, and 1990 to till today, we say the pharmaceutical care era. Before that, it was the India, I mean, before 1940, India was full of this quack medicines, quack pharmacies, doctors, and many others. And that's why the education started. And then this act passed, the drug act passed to prevent the manufacturing sale distributions of substandard medicines. So quality of the medicines come into the picture through this the drug act and cosmetic rules. And then this profession also controlled by this Pharmacy Act. Then 1950-60, we see that this the lot of drug products are produced by the multinationals, but quality come into the picture 1960s when the few incidents or the tragedy took place, particularly thalidomide incidents. You know that this in Germany and different parts of this world, this thalidomide case, thalidomide is a hypnotic drug, sedative drug, which is consumed by these pregnant women to prevent this uneasiness, pain, and other things. But as a result, the baby is born is deformed baby and phocomelia, which is called, and these babies, these children are invalid. You can see that how massacre, disaster in the pharmacy. And normally the family have 1 or 2 child, and they born invalid. What about the status of the family and the parents? Then this question comes to this, the pharmaceutical scientists, the many countries at the time, they seriously investigate the case, and they found that these drugs used is not rationally selected, or its delivery systems was problematic. So there is a adverse drug reactions, and the formulation, the proper formulations of the rational drugs, these all things are coming into the pictures. The whole delivery systems are modified. The selection of the drug based on its physicochemical property, biopharmaceutical property, and pharmacokinetic consideration, pharmacodynamic factors are included in these formulations. Before that, what was there? It was only the traditional formulations, or we can see the immediate release formulations. In this immediate release formulation, there is no at a time, that's totally released, and it's available in the systemic circulations. As a result, it comes to this toxic level and toxic or adverse effect produced. So this has been controlled through these incidents, and then this whole biopharmaceutical and pharmacokinetic pharmacodynamics come into the picture, that how the formulation can be designed, then the drugs will give the maximum therapeutic effect and less adverse effect. So that's why the efficacy and safety, these two are the prime objective of any drug delivery systems. Efficacy, particularly the therapeutic effect, should be the maximum, but at the same time, the safety of the patients or the minimum adverse effect is the basic objective of any drug delivery system. And that is why from 60 onwards, there is a tremendous change in the drug delivery systems. The 1970s onwards, we found this, the microparticle, the sustained release, control release, prolonged release, extended release, modified release, the different type of formulations, spanules, and these microparticles come into the picture. And then the 1980 onwards, this other delivery systems, particularly transdermals and other products, sustained release products and control release products have developed. And as this is 1990, there's a lot of information technology, biotechnology, then pharmaceutical technology developed. Then this more and more demand of more effective the drug delivery systems. And in this way, it is found that this the individual drug particle, the more small drug particles, which ultimately change the biodistributions and change these pharmacokinetic parameters, and ultimately give this very good effect. And so in that way, this nanoparticle, very small particles come into the picture. Later on, we found that this some diseases, as is mentioned earlier, that cancer or many other diseases where the disease is located in a particular area or particular organ of the body, like in the brain, liver, or kidney, or nerves, or some other places. So in such cases, the targeted drug delivery system also developed. So this nanotechnology-based drug delivery system, including the controlled drug delivery system or sustained drug delivery systems or targeted drug delivery system, is the present day need. And I think this maximum work is done on this one. And we got this effect also. There are some products already come in the market, nanotechnology based products and this control release products. So this introduction, this nanotechnology based drug delivery systems will go through it. This is a why we need this new drug delivery systems. You can see here that this the ultimately with these nanomedicines or individualized medicines, and we need this for that we need this controlled drug delivery systems. So this controlled drug delivery systems or individualized medicines means the medicine specifically required to a particular persons who have a special this physiology, pharmacology, and all these systems. So for a individual patients, what drug is required and what it will be its dose. If that nanomedicines is the ultimate goal, then we have to consider this the controlled delivery systems, and then how the drug deliver in a controlled manner. And for this controlled delivery systems, what we need that fundamentals, so particularly the biopharmaceutical features and different routes, different methods, and biopharmaceutics, pharmacokinetics, physicochemical properties, and all these things. So they are the fundamental things which ultimately help for a better drug delivery systems. Well, drug delivery, it's the appropriate administration of the drug through these various routes in the body for this purpose of improving the health. Now that is the basic objective of this drug delivery. It is highly interdisciplinary. Interdisciplinary means the drug, when it is considered as a pure form, which will give this maximum effect. So chemistry is required. It's a product is necessary. That means the pharmaceutics, chemistry, pharmacology, and pharmacognosy, like in the different things, and as well as this biochemistry, microbiology. So all these subjects are this interdisciplinary. It is not a young field, of course. Last say, as you say, that around 70 years in this field of the drug delivery system, 70 or 80 years. It has recently evolved to taken into considerations, particularly the drug physicochemical properties of the drug and these body effects, and in the interactions, improvement of drug effect, and this patient comfort and well-being. Now, so this is the basic objective of controlled drug delivery systems without considerations its physicochemical properties and pharmacokinetic properties and this pharmacology and this patient-related factors, right? Patient compliance or patient comfort, well-being of the patients, and many other things that come into pictures which are covered in the pharmacy practice. So these are the beneficial effect of this the drug delivery systems. This is a common picture, as you know, that when you compare the drug delivery systems, obviously this immediate release or traditional release or sustained release and control release. So these are this being compared, and as you know that this there is for a particular drug there is a therapeutic range or therapeutic window, and if the drug are available in the therapeutic window, obviously it will not give the toxic effects or no, I mean, subtherapeutic effect that can be avoided. So our basic objective in the drug delivery systems is to maintain the plasma drug concentrations in the therapeutic window, right? And if it is a uncontrolled manner or without rationality, the drug is delivered, particularly the multi-dose systems, so it will go ultimately to this toxic level, which is an unsuccessful therapy. But we have to make it in such a manner that it should remain within this therapeutic range, and that's the successful therapy. There are different technologies are there, like This nanoparticles, you say that these particles are having at least this one dimension in the range of this 1 to 100 nanometers. The nanometer I am coming, this nanometer, 1 billionth of a meter, 10 to the minus 9 meter. Then green chemistry, which will be discussed a lot in this FDP program, particularly the green synthesis of this nanoparticle and its the targeting. So what is green chemistry? It is a utilization of set of principles that reduces or eliminates the use of this generation of hazardous substances in the design and manufacture and applications of this chemical products. That this hazardous or this the toxic effect of these chemicals during this manufacturing of these nanoparticles. So that's an important issue. I'll come in. Similarly, the green synthesis of nanoparticles, this is also use of the biological routes, such as those involved in this microorganism routes or plant products, right, for the synthesis of these nanoparticles. My area is, I'm not going to discuss this much more about this, the green synthesis of nanoparticles. I'll discuss this nanoparticles and the drug delivery systems is important and other things. The nanoparticles are other that I think Dr. Das and other speakers will speak about. Little about the nanotechnology based drug delivery systems here. This nanotechnology and this it's a major applications and this why the nanoparticles need and different examples will give you. Everybody know about nanoparticle. Need not to say much about this. If you look into this whole wall, which is a 10 million times smaller, that is a football, of course, but it's a 1.27 into 10 to the power 7 meter. That is the this whole globe, our Earth. And if we compare this football also, and from on the football size 0.22 meter, then it comes to this fluorine, which is this nanoparticles, which is 1 billion times the smaller or 10 to the minus 9 meter. So such a small the particles, right? When the larger particle, as you know, in the physics or physico chemical systems, when a particle becomes the small and the smaller, then its a surface property and its characteristics changes. Particularly in case of this is the nanoparticles, right? Nanoparticle, nano-size particles. It is its properties are quite different from this the macroparticles, which normally we deal the particles or the macromolecules. The small particles, you know, particularly in our drug delivery systems, it can easily pass through this our biomembranes, and its biodistribution is far more, and its bioavailability also much more than this the macroparticles or microparticles. And that's the difference between the microparticles and these nanoparticles. The microcapsules and nanocapsules, too, if you compare, then obviously this nanoparticles, it has this better bioavailability and therapeutic efficacy. There are some examples are there in terms of this the nanometers, from water to this the tennis ball, in terms of this nanometers, right? All these advantages, there are many more advantages. That is why this nanotechnology or nanoparticles or nanotechnology based drug delivery systems are important or advantageous. First thing is, it is a smaller size. As you say that due to its very small size, its biodistributions and its bioavailability is far better. Because of its high surface area, as this bigger particle reduces in the small sizes, the surface area enormously increased. Higher bioavailability, as it is easily cross these biomembranes, and it partitioned easily. So it comes to this very easily to our blood or circulation systems. So its viability is better. Low dose required, obviously, if the viability better means the dose can be reduced, uniform delivery of the drug, right? And it can be administered via these different routes also, either oral or parenteral, or even other routes, even the topical routes, the transdermal and other routes it can be given also. Ability to incorporate this hydrophilic and hydrophobic drugs. As you know that this solubility and permeability of the drug is a very important issue. So any hydrophilic drug or hydrophobic, both category of drugs can be given, right? In case of these liposomes, as you know that this in this liposome, this is both hydrophilic and hydrophobic drugs. It can be even niosomes also. This both category of the drug can be delivered. So it is suitable for the different properties or different category of drugs can be delivered in this manner. Longer shelf stability, the stability of the drug in the biological systems is much more. High carrier capacity. These nanoparticles being the good carriers, drug carriers, right? Drug can be carried from the site of administration to the site of actions. Its performance and efficiency is much more. It's a targeted delivery of the drug at the cellular and this the nuclear level, right? The drug targeting is another the very large area where the drug is targeted to a specific organ cells or to the site, right? So particularly it is applicable for these cancers and some diseases where the drug should be delivered to a specific organ without affecting the normal or healthy cells. So this is the basic objective of targeted delivery systems. Longer clearance times, if it is a controlled delivery systems, it's a elimination half-life and biological half-life can be altered so that the long residence in the biological systems so that it gives the prolonged effect. The ability to maintain this release of the drug, sustain the release of the drug. Like as I say that the preliminary or first daily or in a traditional manner, rather the drug release should be sustained. And then certain disadvantages are there. I mean, coming. Yes. So these are the many advantages are there, but at the same time, there are many hazards and the risk of these nanoparticles are there. Now, what are those hazards? Because this becomes important, particularly in this the FDP program, as is green chemistry and green synthetic chemistry of nanoparticles will be discussed. So this effect of this inhaled nanoparticles in the body may include this lung inflammations and the heart problems. What are those? Heart problems and lung inflammations. Hazards include increased production of this reactive oxygen species, ROS. This reactive oxygen species, it causes the damage. What are these? The DNA damage, genotoxic effects, damage to these organs and tissues in humans, effects on this growth and yield of this crop plants, negative impact on this beneficial bacteria in the environment. So these nanoparticles not only adversely affect in the body, particularly in the organs, but also in the environment and in the plants and other things. So that is why these hazards and risk of nanoparticles should be taken into considerations, right? Because of benefit and advantage, we are abruptly using the nanoparticles in the delivery systems or for its effectiveness. But at the same time, how far it is safe, that also to be considered, right? Other things, the nanoparticles have a high tendency to generate these excessive amounts of the reactive oxygen species, as they say, but due to the strong oxidation potential, the excess ROS induced by nanoparticles can result in the damage of these biomolecules and organelle structures and lead to these protein oxidations, carboxylations, lipid peroxidations, DNA or RNA breakage, and membrane structure destructions, which further cause this necrosis, apoptosis, or even this mutagenesis. See, such type of fatal effects, where the cell deaths or cell damage takes place due to this nanoparticles, which ultimately produce this reactive oxygen species. So this matter should be considered, right? So this is the hazard, and that is why the natural way or this green chemistry is important one. Well, this let us discuss about this synthesis of these nanoparticles. Basically, the nanoparticles are synthesized by two technology. One is that top-down methods and bottom-up methods. Right? In the top-down and bottom-up method, now this bottom-up from this bottom, from low level to this high level. That means very small molecules to these higher molecules, right? It's like the synthetic, right? In bottom-up, the synthesis. In bottom-up synthesis, there's the nanoparticles can build from the small entities. For example, by joining these molecules and the small particles. The small molecules, as you know, the proteins is synthesized from these amino acids or fat from these fatty acids. So similarly, in a bottom-up method, these small molecules, so they join together and form the bigger these nanoparticle sizes, and where if the drug particles are there, I mean this polymer are synthesized and give the coating or I mean the structures which incorporate the drug, right? In case of this the reservoir systems or in a matrix systems, in case of this reservoir systems, as you know that this the polymer molecules they synthesized and forms a coating on this the drug particles, and in the core the drug molecules will be there, and in the coating that the polymers will be there. So similar and in case of this matrix systems, the polymer, the structure is developed and where the drug molecules are incorporated. So in a bottom-up method, the process is developing like this. In a bottom-up synthesis, this the macromolecules, the building blocks of these nanoparticles are formed first and then assembled to produce these final nanoparticles. And this bottom-up synthesis, the molecules relies on this chemical and this biological method of productions, right? So this is the bottom-up method, but this is somewhat limited in the chemistry and other cases. But in all pharmaceuticals, the mostly this top-down technique is used. Top-down techniques, here the top-down synthesis, these nanoparticles are produced by this size reductions from suitable starting materials. So first of all, this the bigger materials are produced, and then by the size reduction, these small, I mean, these nano size particles are produced. So the size reduction is achieved by these various physical, chemical, or mechanical methods. The different techniques, for this nanoparticle, it is just like an microparticles, right? Ionic gelations or this co-adsorption phase separations, or even these other methods. Lot of many other methods can be used for this production of these nanoparticles. This top-down production methods that introduce the imperfections in the surface structures of the product, and this is the major limitations because of this surface chemistry and this other physical properties of the nanoparticles, and are highly dependent on the surface structures. You know, as these nanoparticles are this control release products, and drug from the core is to be released through this the coating film, right? And this around these drug particles or core material, the polymeric film should be thin and uniform, and uniformly the porous, so that drugs can be released through this the pore structures. Of course, there are different release mechanisms are there, right? Dissolutions or diffusions or swelling or erosions. So different techniques are there. So, so all these things are done through this polymeric films, which ultimately control the release. So this structure of this film or this topography, surface topography, is a very important one, right? In case of this top-down method, this is to be controlled. And that is why we have to observe it under the scanning electron microscopy and see this porous structure, or even the atomic force microscopy is used to see this how the surface structure is developed for this purpose. There are different characterizations or property studies there, because without characterizations, you cannot know that this how waste it will give this effect, or how waste it is produced also. So the nanoparticles are generally characterized by their size, shape, and the surface area, and thus the dispersity. Size is important. As we say that it is a nanoparticle, it should be in the nano range. But nano range means for base therapeutic effect, it's a 50 to 100 nanometer is good. 150 to 100, but it's more than that, right? Even again, it gives towards the like this microparticles. So around 100, it's a good size, right? And shape, obviously, the spherical shape is an important one, so that this the coating films given on this core material, and this release can be controlled, or release can be predetermined also. So this spherical shape is an important one, and to make this spherical one. Surface area, obviously this how much surface area is increased. That is also see when the surface area is increasing, the surface free energy and the zeta potential. That is this is important one, and that leads to this dispersity, particularly in case of this nanoparticle, the suspension or nanoparticle emulsions. This dispersity or agglomerations are very important issue. That's why the polydispersity index and other things, this is also considered. And this that is why the zeta sizer is used to determine the molecular size or particle size. Then this surface potential, zeta potential, and PDI, polydispersity index, and all these things are characterized through this the zeta sizer, and this scanning electron microscopy and other things are used to determine the size. Other things, the homogeneity of the properties is important in this many applications. Homogeneity, obviously, the small size, big size, if there are a lot of variations, are their properties will be the variations. So in that case, the effect also will vary. The common technique of characterization nanoparticles are like EV spectrophotometry. EV spectrophotometry, obviously, the amount of drug incorporated or the encapsulations to estimate that this obviously good quality, this EV visible spectrophotometer required. And then this dynamic light scattering microscopy, then and this gives the size and characteristics of nanoparticles. Scanning electron microscopy, this also this particularly for surface characteristics, how the surface is smooth or the porous structures and all these things. And that's why the ACM or scanning electron microscopy, then the dispersive spectroscopy, transmission electron microscopy, then Fourier transform, infrared spectroscopy, and X-ray diffraction. So these are the different type of very advanced equipments are used to characterize these nanoparticles. Whether there is some drug polymer interactions, FDAR, or if the crystal drugs are there, drugs are crystals. So in that case, then in the change in the behavior of the crystals, the XRD. So like that, these the different modern techniques or with these advanced equipments, these are used. Okay. Innovative applications. There are a lot of applications. I think you know I am not going through it. Now let us see some challenges about these nanoparticles. This the formulation, stability, and the shelf life, right? When a very fine drug distribution of fine particles, particular drug distributions, and the products and in our biological systems, you have to see that this it's not reacting and whether it's maintain the stability, because biological environment in our body, this is different. pH in the GIT is different in the different segments. In the enzymatic systems are there. In the blood, there are other things are there. So this the stability of the drug is important one, and maintenance the shelf life, that is also very challenging task, right? So that you can give this a prolonged effect. Prevention of the drug from these biological degradations, right? So every possibility, either the first effect or anywhere these biotransformations or any interactions with these other things, with the drugs or biological components, which ultimately lead to this the degradations or decompositions, that has to be prevented. And that's why here the drugs are protected by this polymer, and the selection of this polymer is a very important one, so that this polymer coating ultimately, it's a carrier, it will ultimately give the protection to the drug and reach to this site of effect. The effective targeting, when the drug targeting required to a specific organ, like in a cancer, in the brain or liver or kidney or heart, or even in the nerves or other specific organs. So it's a effective targeting. So there are different method of targeting are there, but you see that is ultimately the drug targeted to the organ, and particularly the cells or organs, not adversely affecting the normal cells, right? Because if the normal cells are damaged, then this no use, no benefit, rather it has this more damage or adverse effects. Patient compliance, yes, today in this age, the patient needs comfort in the drug administrations, right? Either once in a day, twice in a day, or in a nasal way, or in a buccal way, or in a topical way, or in the skin, whatever maybe. It should be very comfortable, right? Not very painful or not need much expertise for this drug administration. So that's why the patient compliance is an important issue. Cost effectiveness, yes. Today, the cost of the drug is escalating, and it's very high also. So if you more amount of drug required, then this cost is high. At the same time, it's a damage also to the body, because more and more few drugs are used, more than this your therapeutic dose, then obviously the adverse effect will be produced. So in case of this nanoparticles or nanotechnology, it is calculated based on this biopharmaceutical calculations or pharmacokinetic parameter study, that this exact amount of drug can be given through this loading dose and maintenance dose. And that's why the cost is less, because the exact amount of drug required administered. And moreover, the other effect is the less time to stay in the hospitals, right? So expenditure in the for therapy in the hospitals and other places. So that can be the reduced. Product life extension, yes. Since the drug is protected, either encapsulated or in a matrix form, so drug, the life is extended, right? So these are the challenges. One being a nanotechnology scientist or technologist, he has to look all this physical-chemical, biological, biopharmaceutical, pharmacokinetic, pharmacodynamic aspects before going for preparation of these nanoparticles. Yes, there is some application of uh, this nanotechnology in the day-to-day life. I think it is being discussed or it will be discussed thoroughly. This in the, there's a lot of things are done in the information technology or in the energy sectors. And other than this, our medicines, particularly the cancer treatment, bone treatment, this DNA, drug delivery systems and drug development, and these medical tools, diagnostic tools, and this imaging technology, all these things in the medicals and in the computer tools, obviously. Other than this pharmaceuticals, this nanotechnology used in the chemistry, physics, and many other areas also. That's why today we talk a lot about this, the nanotechnology, nanoparticles, and all these things are there. Challenges to make the drug more effective by this nanotechnology. The prevention of the drug from this biological issue, already I told you, right? In the cancer therapy also, the diagnosis using this quantum dots or tumor targeted delivery, imaging, or the cancer gene therapy, these are the very recent area. This is some scenario, this how this improving this cancer treatments, this how these cancer cells and the other normal cells in case of this the nanotechnology based drug delivery systems, the non-cancerous cells, here in normal case, traditional treatment, it's affected. The cancer cells as well as the normal cells both are affected, right? And this damage both. So including this, the particular target, organs affected by these cancers. So this whole organ will be damaged. Suppose you have a brain cancer or cancer in the pancreas or cancer in the uterus, right? Or in the breast cancer. So beside this, in the traditional treatment, this whole organ will be damaged. Then what is the benefit? In compared to this, in nanotechnology, these cancer cells are affected. It can be killed, right? These nanoparticles and its release drugs. But whether the other normal cells, they are protected, right? They will function as usual. So that's why this nanotechnology used, and particularly here the targeted delivery systems is used. And that's why its demand is very high, right? It can kill the cancer cells in the particular organs. Yes, these are the dead cells. Other nanoparticles for drug delivery systems, these metallic nanoparticles, this is the very common metal-based these nanoparticles. The different type of metals are used. Lipid-based nanoparticles, these liposomes are niosomes. These are the used. Polymer-based nanoparticles, right? Rendimers and these micelle, different type of micelles. Biological nanoparticles, like the bovine, albumin, they are used. Let me say this a little bit details. This liposome nanoparticles, this liposomes made of phospholipid, cholesterols, and other things. It's a bilayer structures, right? And in a inner core and outside, this is two zones are there, even this inside also. So drug based on this nature, either if it is a hydrophilic course, the hydrophilic drugs can be entrapped here. And but if it is a lipophilic, then it will lipophilic drugs will be here. So it's both hydrophilic and lipophilic drugs. And since it is a natural products, I mean, obtain this cholesterol or phospholipids, they are the biocomponents. So it's a very biocompatible and it's a very suitable, but it's a stability and other problems are there. Liposomal circulatory systems, you can see here, the different type of materials can be delivered, right? And in case of this drug targeting, there are different type of the targeting ligands can be attached here, right? And these ligands, it can carry this carrier as a whole, this drug carrier, these ligands, it will take these materials, I mean, this nanoparticle or liposome to a specific organ. So this monoclonal antibody is selected from a particular organ, which is very specific to that organ, that will carry the drug molecules, I mean, these nanoparticles to that site, and it will release there. And then the drug will be released from these liposomes. And these liposomes, I mean, other these materials, this will be degraded and it will not affect much in the body because these are the our biocomponents, and released drugs will entry into this the cell, and as this EPF effect and others, as this say, the enhance retention permeability. So this EPR effect, I mean, the drugs enter and give this effect, right? Niosomes made of this non-ionic surfactants. It is similar to this liposomes, and yeah, here also the drug can be encapsulated and drug can be incorporated, and it can be also in necessary cases it can be this also targeted, right? Targeted with this the niosomes, and it can be delivered also. So this there are different zones are shown here, hydrophilic and hydrophobic zones. So this hydrophilic and hydrophobic both category of drugs can be incorporated based on this synthesis and structures, right? So what type of drugs you are selected, and accordingly these liposomes and niosomes can be synthesized or prepared, and it can be delivered also. Polymer based this nanotechnology, you know, today this polymer, in fact, the key factor or instrumental factor in this nanotechnology or in this drug delivery systems is the polymer. The role of polymer is a tremendous, you know, and the polymer today in the different category of polymers, either hydrophilic or hydrophobic or natural or synthetic or semi-synthetic polymers, or mucoadhesive polymers, or this bioadhesive, biodegradable. There are different category of polymers are available. So these polymers are used for this the micelles or this the nanoparticles. You know, this this micellar formations in this niosomes and other things, that technology used here. Dendrimers is the other category here, you know, the itself in one structures, there are polymeric structures, many more drug molecules can be incorporated, right? It's a very high degree of this molecular uniformity, right? And this narrow molecular weight distributions and many advantages are there. This is the reason that dendrimers, polymeric nanoparticles, as you say, that you can see that this the different type of these structures. There are network structure, cross-link structure. This is a like actic structures, and the core is coated with these polymers, right? It's a and the capsules. So either it's a reservoir systems or monolithic or this matrix systems of these nanoparticles, very useful for this drug delivery system and control the release of the drug also. Polymer nanoparticles, so this already I have mentioned, carbon 60 fluorines, this is a other one. There are a lot of cages and lot of spaces created here for incorporating the drugs. Similarly, this carbon nanotubes, they are the, again, this the tube like the structures where drugs can be incorporated in the different manner, right? Biological nanoparticles, where this the serum albumins and other things are used here. Not much discussing. Drug carriers, drug delivery carriers. These are already the basicals like liposome niosomes and pharmaceutical carriers. This the micellar structures. Then this liquid crystals, nanocrystals, and nanocapsules, and these other materials, these nanospheres. So even this the multifunctional, the dendrite nanoparticles. So different category of class of these nanoparticles are here. Significant achievements of this nanotechnology so far: development of this 1 dose a day, this process, this drug, but this many drugs are used, even anti-cancer drugs are used also. Once in a dose, yes, this is an achievement, where in a traditional way that drugs has to be administered the 3-4 times or more than 1 times, but here the frequency of administration can be reduced, dose can be reduced also. Tumor targeted of taxol delivery, this is a major advantage. Improved ophthalmic delivery formulations using this smart hydrogel nanoparticles. These hydrogel nanoparticles can be used not only ophthalmic, but the other oral mucosa or I mean, even also in the nasal, buccal, vaginal, many other cases, these hydrogels and nanoparticles can be used. Oral insulin formulations using these nanoparticle carriers. You see here, the insulin being a protein, the macromolecules, the orally cannot be administered, but when it is given in a nanoparticular form in a nasal and other way, these the macromolecules can be absorbed directly into these systems. So that's also possible, the advantage. Liposomal based doxorubicin B formulation, this is already available from I think 90s. So this is a big achievement for this breast cancer and other things. This doxorubicin, these liposomal nanoparticles. So it, last I should say that it involves this low cost research compared to that for delivery of the new chemical entities. If you look into this overall cases, then obviously in safety and efficacy of the drugs are considered, then this is the best one. Minimizing the drug usage and would significantly reduce the effect cost of this drug, which would give this financial benefit to these patients. This is a, today the need or need of the hour is not only reduce this side effects or adverse effects, it also reduce the cost. So cost effectiveness is a very important issue. Cost effectiveness, patient compatibility, then the I mean pharmacovigilance, which is that ultimately that adverse drug effects and all these things, they become the issue. So these issues are addressed by this nanotechnology, right? Today, what you need for pharmacy practice area. So that rational drug therapy or effectiveness or cost, patient compliance. So all these things are possible through using this technology. Novel means the delivery, particularly using these nano carriers, can allow this the branded drug to the rescued from this I pass this genetic competitions. Of course, there are other problems are there. This is my first part, obviously. So here we just mentioned this what is the nanotechnology and the drug delivery system, particularly the drug delivery systems, how it is important, how it is potential, how it is essential also in the present day for better therapy or effectiveness and the safety cases, right? Everybody, particularly our regulatory affairs, they are very particular when approving the drug particles to see its safety and efficacy. So this is possible if we judiciously calculate and implement this nanotechnology in our drug delivery systems. So the drug delivery is a very important one. Nanotechnology also very potential technology and very advanced technology. If both are combined together, then ultimately it will be beneficial for this control and management of many diseases which are uncontrolled earlier, and it is also beneficial for human society particularly, and it increase the lifespan, the lifespan of this the common people. You know, this earlier days, the people, the self life, our day by day increasing because our healthcare system is improving. Our recent drugs and delivered drugs, there are more potential and more effective, and that is why our disease is controlled in a better manner. Diseases are managed and people live very happily with less pain than others. So that's the blessings, right, in the present generations or present society from this nanotechnology and the drug delivery systems. Okay. Well, if you have question, then I'll go some work. Obviously, we work on this nanotechnology and Buddhist and nanoparticles. We can discuss a little bit, and then yes, madam.
Sir, shall we start the question and answer session?
If you allow this, I'll just show some slides.
Yeah.
Few slides on this my research area.
Yes, sir. Sure.
Right.
Yes, sir.
And then we can take up this questions.
Yes, sir.
Can you see the slide?
Yes, sir.
So this is a preparation of this budesonide loaded these nanoparticles and effect of the different stabilizers. We prepared it is in the Jazan when I was in the Jazan University, right? And so that work done by my on students, so Dinda, right? And so aims and objectives, as you know that this the small particles or nanoparticles are produced here. I'm not going all this all these things, right? Budesonide drug, you know, this very useful drug. This method of preparations, how these nanoparticles are prepared in a schematic way, is given here. So in this, you know, the normally the nanoparticle preparations, as these very small particles, and if that nanocapsules are there, the drug particles should be encapsulated properly. That means the coating should be done in a proper manner. If the improper coating, the drug will be released. It is a improperly used. So that's why it should be properly coated. And that's why this nanoparticle preparation or manufacturing, there are a lot of formulation parameters and processing factors are considered, right? So that is done here. I'm not going much about it. See this characterization of these nanoparticles. by this scanning electron microscopy. This is the freeze drying product PLG nanoparticles with budesonide, right? And in the scanning electron microscopy pictures. This is the different, the different stabilizers are used in the preparations, PVA or carbopol or polymers. So these are the different components used in the preparations, and during homogenizations, different processing parameters, right? And this different processing parameters and formulation parameters are considered. It can be optimized also. Then this is your atomic force microscopy pictures. Here the picture shows the characteristics of this coating, how it is developed, right? Again, the drug loading, encapsulation efficiency of this, the different formulations are done here. The pressures in the different pressures, how it affect these formulations. So that is see this micro encapsulation efficiency and the size of nanoparticles. So that are studied here and presented here. This drug release characteristics, in vitro drug release, right? Some pharmacokinetic study also is carried out, right? The in vivo pharmacokinetic study and biodistribution pattern, and this how it changes for pure drug and budesonide loaded PLG nanoparticles. So how is this area under the curve changes, and how this clearance value also changes, and then this half-life also, you can see that half-life from pure drug 6.81 to this 12.47 is a great achievements of this nanoparticle. And mean residence time also, this has been changed. You see this in case of pure drug where it is 9.8 is 17.99, it's almost that double, and half-life also this double. So it can give this prolonged effect, I mean to say control effect, right? Control release effect. So this with this the frequency of administration can be reduced also. This is other imaging things. So this is the conclusion. Budesonide loaded nanoparticles can be successfully prepared with this high pressure emulsifications and solvent evaporation technique. There are almost the 3 stabilizers used in this formulation. PVA found to be the best one, and this higher the pressure as well as this the number of cycles applied, the smaller the size of nanoparticles produced. So these nanoparticles can be produced, but lot of care and the lot of optimization are required to produce a small particles, but with a desired release characteristics or desired targeting property, right? It needs a lot of skill, lot of science, and lot of factors. So that's why your selection of drug, from selection of drug to selection of polymer, selection of method, and the production technology, characterizations, in vitro and in vivo study, all these things, if they are proper, then we get these nanoparticles, and which will be very effective ones, right? And if you can get this all this biological study, right, like pharmacokinetic parameter studies, then you can achieve that this your nanoparticle is not producing that hazards, I mean not adverse effects. So this is the ultimate objective, that yes, we are getting benefit, but at the same time, we reduce its adverse effect, which ultimately cause hazards to the body, to the environment, to our whole systems. And that's why comes the green nanoparticles. So this before your green synthetic nanoparticles, this is that potential advantage or effect of these nanoparticles, right? If it is not prepared or used in a controlled manner, then obviously it produces the hazards. And the second step, your this green or green synthetic nanoparticles required. So with this, I thank you, all of you,